So in genetics, now we are going ahead for the lab techniques which are very much used in genetics. So the first one is the blotting. So regarding the blotting techniques, these are very much tested, very much important, and we have a mnemonic, very common mnemonic for this, that snowdrop. And snowdrop stands for southern blotting, northern blotting, western blotting, and southern blotting detect DNA, and northern blotting detect RNA, and western blotting detect protein. So how this blotting technique actually work, that first of all, the steps which are common in all of the techniques. The first step is we take sample, and this sample is undergone. If this sample is react with or enzymatically cleaved by restriction endonuclease enzyme, enzymatic cleavage. This sample is anything. This sample is our target of interest. If we are going to detect protein, the sample may be protein. If we are detecting messenger RNA or RNA or DNA, the sample is vice versa. So first sample is first sample is cleaved by endonucleases. Then we do gel electrophoresis to separate these cleaved parts of the sample, because whenever the sample is cleaved, it has different parts, and these parts are separated by gel electrophoresis machine. After separation, then now we label that different parts. So for label we use filter nitrocellulose, which is used for the display of the sample and also labeling. So now we, after separate that electrophoretic segments, now we do radioactive probes which highlight and recognize the different parts of them, which recognize different segments. After recognizing, now we film those particles and visualize as lab, visualize or label or visualize a sample. So overall, summarize this process. First, the sample, which may be either DNA, either RNA, either a protein. A sample is enzymatically cleaved into small proteins into different parts by endonucleases enzyme. Then we separate them by a gel electrophoresis machine and transfer to the filter. The filter is exposed to radioactive probes, which may be RNA, which may be RNA binding, which may be DNA binding, which may be protein binding. So these probes recognize and anneal to complementary to the respective samples, which results into labeled piece of the sample which is visualized when it is visualized on the film. So film is used to visualize. Radioactive probe recognize the complementary strands of the DNA of the or the RNA strands. Filter is used to just express those and label. Filter is used to just express those parts, and then we have electrophoresis for separation. So this is the whole general mechanism of all the blotting technique. If we using southern blot, we are dealing with the DNA sample, and it in order the radioactive probe is complementary to that DNA. In northern technique, northern blotting, the sample is process is pretty much simple, but the sample is RNA sample. It may be messenger RNA, which is electrophoresed, and then it is useful for studying messenger RNA study. So northern blot is for messenger RNA studying, which is reflective of gene expression, obviously. Then we use western blot. In the western blot, we are detecting the proteins, which may be an antibody, which may be anything. So there is a different comparison between a western blot and ELISA. What is the difference? That western blot first separate protein and then detect and then visualize or then detect the rest of the things. So western blot is using gel electrophoresis, all the membranes and the label different proteins. So there is a difference from western blot and ELISA, that western blot first separate the proteins, whereas ELISA it directly detect proteins from the serum. So this is a difference. Rest of the technique is very much similar. ELISA and western blotting are very much similar, but this is the difference, that western blot is pretty much separate the protein first, then detect them, but ELISA directly detect from the patient serum. And then we have the very much special blotting, which is southern western blotting. And as the name indicate, that southern blotting, they are detecting something which is DNA and which is western protein. So they are the proteins which directly bind to DNA or DNA binding protein. So southern western blotting are the DNA binding proteins. For example, what are the DNA binding proteins? The transcription factors, these all are DNA binding protein. Then we have the histone, these are also DNA binding proteins. Then we have the endonucleases, these are also DNA binding proteins. And in the example for DNA binding protein, there is a motif which is very much common, which is known as the C-Jun or C-Phos. These are the leucine which is present in nucleotide. Leucine is the protein. So leucine zipper motif. So these are the J-Phos and C-Jun or C-Phos are the DNA binding protein, DNA labeling protein, which are actually the leucine zipper motifs, and they directly bind to the DNA segment. So southern western blotting is a technique which directly detect the proteins which are DNA binding. So this is all about the blotting technique, the mechanism and the different comparisons.